Very much, ladies and gentlemen. On behalf of Shim-Pol A.M. Borzymowski, I would like to present you a very short presentation about solutions in biopharmaceutical biotransformations and metabolomics, and I hope this will pique your interest. In fact, PEEK as a material is very useful in bioanalysis, and I will talk about this among others. In April 2025, our company will celebrate a very nice 150 years anniversary. So it's a quite interesting and quite long story of our company. And from the beginning, the corporate philosophy was to contributing to society through science and technology. And here we have our founders, Genzo Shimadzu Jr. And his words, science is a practical endeavor. There is no point in theoretical knowledge if it isn't applied to help people. And here you see our logo of our anniversary. Maybe it's not so visible, but here we have a balloon. It's because at that time it was very popular to start to launch a balloon in Europe and also in Asia. And here we have Japan's first successful manned balloon launch. It was really it was very important for Asians, for Japanese to do such activities. It was in Kyoto in 1877. Just to compare, it was also very popular in Europe. In Warsaw during Stanisław August Poniatowski times, we also launched the balloon in 1789. So it much many years earlier. I couldn't find the proper picture from Warsaw, so here I can present the photo from Haga from 1785. We had many interesting anniversaries last times. Among others, we celebrated 50 years of mass spectrometry innovation in Shimadzu, but also 100 years of innovation in testing machines, but also in analytical balances. Not so many people know that Shimadzu is also producer of fantastic balances. But we had also anniversary 115 years of innovation in the X-ray systems from Shimadzu. So that's why we can provide you the total solution in pharmaceutical industry, but also in food industry, for food market, also in biotechnology, manufacturing, but also in health. So we entering the diagnostic market. We are offering a lot of interesting solutions, among other next generation genetic testing, cell assay technology, microbial high-speed screening technology with multi-TOF technology. We are the only one company on the market with autonomous laboratory systems without human being inside, with the application of artificial intelligence, etc., etc. Our history of liquid chromatography is very long, but the latest systems, bioinert systems, comes from 2022. Here we have our latest product with many patents, among others enhanced optical performance, patented autosampler with ultra-low carryover, dry airflow control, ultra-fast analysis cycle time, auto-diagnostic and recovery intelligence. We have also eco-label because of very small dimensions of the systems and very small electricity consumption. Here we can see trends of biotherapeutics, and we can observe big growth of this segment. When we are talking about application requirements, we can see that we have to apply many techniques, among others LC-Sec, for example, for aggregate formation, or ion chromatography for methionine isomer, or LCMS, or combination of ion chromatography and MS, for example, for analysis of lysine isomer. Ion exchange chromatography for aspartic acid isomer, or for example for peptide mapping reverse phase LCMS. On the other side, for glycans, we have to use multi or LCMS systems. For antibody drug conjugation, we can use SEC or ion chromatography or even LCMS systems. Here, these techniques are often used in analysis of antibody drugs and require high salt content and extreme pH conditions. Stainless steel may corrode under these conditions. Another example, nucleic acid drugs possess active sites that interact with metal surface. Oligonucleotide show characteristic of metal coordination, and adsorption shows a bigger impact when using LCMS system. We can define what stands for bio-inert and biocompatible. Biocompatible, a system in which metal parts are resistant to corrosion. It is suitable for mobile phases containing high concentrations of acids, salts, and halogens, typically used for biomolecules. Here we have example MP35N nickel cobalt based alloy, and here we can see the metal ions launched into methanol solution after 1 month of soak time using a small surface sample set. Bio-inert is a system in which is inert to biomolecules, promotes to no interaction even with compounds that exhibit enhancement adsorption to a metal surface, such as biopolymers. The system is also resistant to corrosion. In addition to phosphoric acid compounds, which are famous for metal adsorption, there are a variety of compounds, including those with carboxy groups, and many are found not only in biopharmaceuticals but also in small molecules such as pesticides, chemicals, and foods. When we are talking about effects of metal adsorption, so here we have pigtailing, it reducing sensitivity, smaller peaks also reducing sensitivity, but also the calibration curves are not linear, and additionally we have also a bigger carryover. In bio-inertness, we have requirements: suppression of molecules adsorption to metal, eliminate time-consuming passivation protocols, and countermeasure is a bio-inert sample flow path with no metal parts. In area of biocompatibility, the requirement is to prevent corrosion from salt content or extreme pH and reduce the time and effort of washing. And countermeasure is wetted parts of the system made from corrosion-resistant material. So here we have engineered solution from Shimadzu. Nexarex is inert with extreme pH stable from 1 to 14. PLS tubing, ceramic autosampler needle, diamond-like carbon stator with reinforced peak rotor. And here are our modules. Here we have a controller of the system, and degazer, heart of the system, bioinert pump, metal-free PDA detector. Also inert pump, metal-free autosampler, automated inert flow switching capability, and also very interesting pH monitoring capability. Heart of our system is a fantastic inert pump with PLS tubing. But also, some people says that heart of the system is a column. So also column with stainless steel body, but covered by PEEK or other inert material. And also a very important possibility to exchange the column without any tools. Here we have some columns from Shimadzu, Scepter Claris and Scepter Metal Free. It's really important to have inert columns in bioanalysis. Some initial results. Tetracycline is a representative example of veterinary drugs known to absorb two metals and glass. From these results, the PEEK line column scepter metal-free and metal-free system provides the highest sensitivity for these compounds. Maybe I will show you another examples and comparisons, traditional UHPLC and metal-free UHPLC system. Maybe another examples just to compare tailing factor to 0.07 in traditional metal column and tailing factor 1.35 in metal-free column. Another comparison. When we are looking for the best sensitivity, definitely metal-free system and metal-free column will be the best choice. When we are talking about the highest resolution, also metal-free system will be better than traditional UHPLC. In this area of bioanalysis, we very often offering lab solutions inside biologics. It's advanced data processing software for characterizing oligonucleotide products and impurities. This software was initially produced for LC-MS QTOF systems. We have created a powerful and flexible data processing package to integrate processing and reporting. It's a key part of our end-to-end solution for any synthetic oligonucleotide workflow environment. Our platform is designed to generate high data quality and tools to make a high impact on the user experience, including merged spectrum processing. It's a patented algorithm from DDA MSMS. Originally, this software was prepared for LC-MS QTOF, but now this software is dedicated for many techniques like LC-MS single quad, high-resolution MS, or even multi-TOF systems or digital ion trap multi systems. When we are talking about application, of course, the LC-MS QTOF system is most versatile solution. But my experience is that in bigger laboratories, we can observe, for example, 20 LC-MS single quads and one QTOF system, or on the other hand, many multi-TOF systems, and for example, one digital ion trap. Features of this software is to display the fragment coverage, depiction of the structural formula to predict the formula to component chromatogram display and many others. Another solution from Shimadzu is a C2MAP. It's a cell culture media analysis platform. Ensuring that cell culture media contains a precise balance of components is critical for biopharmaceutical development. Here we have our system and the features. We can at the same time analyze 97 compounds within 17 minutes. And our customers can get ready-to-use method with optimized and validated analytical conditions and tools. Here we have whole system. So in general, it's a LCMS system, but connected online with a special robot, C2MAP. We can use it for CHO cells, iPS cells, EC cells, T cells, etc., etc. Here we have some comparison of time analysis using C2MAP and traditional approach. A very important part of this system is our software trends. We can see on the left side. And on the right side, we have a table of all compounds that we can analyze at the same time. So it's important to say that sugars, amino acids, vitamins, antibiotics can be analyzed at the same time within 17 minutes. At the end, I would like to ask, does the future belongs to universal detectors? From time to time, we are facing the situation when we have to analyze compounds with no UV absorption, or on the other side, with no ionization in MS or pure ionization. In such a situation, sometimes we are thinking that the only one solution is to take the universal detectors. In this group, actually, we have many aerosol-based detectors. There are some examples of these detectors: NQAD, nano-quantity analyte detectors, corona cut charge aerosol detectors, but also ELSD, evaporating light scattering detectors, or NC-LSD, or even MULS, so multi-angle light scattering detectors. When we compare, for example, results from PDA detectors or RID, we can see poor results, but on the other side, ELSD can get us very, very nice results. Just to conclude, here we have the scientific article, in which, because I have no time, I will just show you an abstract. What is the most important parameter in detectors? For sure, sensitivity. So in this paper, authors show us that the best detector is NQAD, little bit less corona cut, and little bit worse ELSD. But sensitivity is not the only one parameter that we have to consider. Robustness is also very important. In this case, ELSD can be the best solution. Corona cut may be a little bit less, and NQAD may be worse. That's why we recommend third generation of ELSD from Shimadzu. Thank you very much for your attention.